Hey guys, it's me again. I just wanted to record a video addressing some questions I've been getting over email, and most of those questions involve the graphs and the data for the experimental summaries. So this is kind of the format you want to be putting your data into. You need to have some of your data be your without treatment data. This is your control data. And if you remember, we didn't replicate these because we were going to replicate across groups. So one group did 3 plates. Then the other group, so here's some other groups. All this data is fake, by the way. I just made this up. But here are some other groups in the class, and they did 1 plate of each in each group. So altogether, in a whole class, we've replicated the control without each of us having to do 3 plates each. Okay, so you got your groups data. Sorry. And then you have the big group of data, and this is your treatment data. Say your treatment could be, our classes did lots of different things. We did pH, Lysol, different kinds of Lysol, etc. So let's just go with this being different kinds of Lysol. I'll just say Lysol for the rest of the video. And then you've got your different molds. So this is one kind of mold, 3 plates with your treatment. So say mold 1 is PCAM. Alright, so we've got 1 plate of PCAM with Lysol, 2nd plate of PCAM with Lysol, 3rd plate of PCAM with Lysol. Then we've done the same thing for, say, A flavus, could be A niger, whatever our other mold is, 3 times. And then we've done the competition of our 2 molds, and this column, or this grouping might be a little confusing. So this column is mold 1, this column is mold 2. So these are 2 different molds growing on the same plate, okay? And we've done that 3 different times. Each of these 3 plates has our treatment, our Lysol. And then we've got 3 plates that just got Lysol, they didn't get any mold. And that was to control for any effect of contamination within our treatment, like is there mold in our Lysol? Maybe our Lysol is not Lysol at all, maybe it's water with mold in it. Or it could also be from outside contamination sources, like if you didn't seal your plates well enough with parafilm, mold could get in through the crack and start growing on the plate. So you want to look out for anything more than 0, basically, in your no mold treatment controls. So that's these red values here. So that could indicate that mold got in on day 5, or maybe that there was some contamination in the treatment, but it didn't begin growing until day 5. Let me recap the way this is set up again, because I missed this part. So you're putting whichever kind of plate it is here, and then these are the different days, and this is the percent cover on that day. And in my hypothetical example, I've said that I counted 10 intersection spots on my plate with the mold present out of 255. I know probably that that's not the correct number for your plates. I just made a number up because that number could vary between different groups, and I need to find a number, so I just picked 255. So you want to make sure that you're using your correct number for each of these things, and you also want to make sure that it's a percentage, because this number might vary. It might vary between the people who were measuring the plates, depending on how you guys measured your plates, whether you took pictures or whether you lined up the transparency. And it could vary between groups, it could vary between classes, so just make sure it's in a percentage, okay? Now the other thing to consider is that if you were to graph all of this information into the same graph, there would be 15 different lines, and that's way too much information. I'm not going to be able to understand, nor you, what is in your graph. So instead, what you need to do is make an average. That's why we've replicated. If you look at mold 1 here, there's quite a lot of variation in the mold 1's growth. These first two plates are not too different from each other, 90% at the end, 98%, but the first plate only got up to 37%. It's very, very low. So something was wrong with this plate, and if we had only done one plate and it happened to be this plate, then our data would be very different than it is now that we've replicated. So 3 is like the bare minimum replication. In a real study, you'd want to do much more than just 3, but that's what we're working with right now. For the control data, we've got a little bit more than 3. So you're going to be averaging your control plates data with the rest of the class's control plate data, but you need to make sure that you're matching plate to plate. So you don't want to average different molds together, because that doesn't make any sense. They're different molds, they're going to have different growth rates. You don't average apples and oranges together. So for my average data table here, if you see the actual formula for that spot is average of my data plus the group number 1's data, group number 2's data, in that same plate, same day, and group number 3's data from that plate from that day. Okay, and I did the same for mold 2 and the competition plates. For the competition, you need to make sure that you're not averaging, again, those two different molds. It needs to be mold 1 with mold 1. Okay. So you get the average control data. That's your without treatment data, again. And that's coming from the entire class. And then you have a with treatment. And with treatment, you run into a similar issue about how you're doing averages. So you don't want to use any class data for this, because each group used a different treatment. You wouldn't average, say, mold 1 growth on Lysol with mold 1 growth on high temperature or high pH, because those effects are going to be very different, and it doesn't really mean anything to put an average of those together. So you want to make sure that this part, the with treatment part, is only your data, because it's your treatment, which is different from everyone else's treatment. So we're going to get a number for this spot, which is mold number 1. What's the average growth on day 1 across the 3 different plates, the 3 different replicates, in other words, that we did? It's the average of these 3 numbers here. Okay, 6, 12, and 2 averages to about 6.5. Alright, and we did the same for 2, averaging across here. We did the same for competition. Now, you have to be real careful here again. You don't want to average this way, because that would be averaging mold 1 and mold 2, apples and oranges. You want to make sure you're averaging mold 1 and mold 1 and mold 1 on the 3 different plates, and mold 2 with mold 2 with mold 2. Okay. And then for the no mold plates, it should be zeros across the board, hopefully. But if you have a plate or 2 plates that showed some contamination, throw those out. Okay. If all your plates had contamination and it was fairly consistent, then you can get an average of contaminant growth by averaging across this way. That might be kind of an interesting thing to see in your report. Okay, now we're on to the actual graphing. So this is where most of the questions come in. How many graphs? What do I graph? That's the kind of things that I'm hearing. So you've got basically 4 different graphs you could do. The first one is control mold growth with and without competition. This is like what we did in part 1 of the lab. You've got your mold 1 growth without competition, mold 2 growth without competition, mold 1 with competition, mold 2 with competition. So you can see how things change whether they are or are not in competition with each other. Okay, And then you can look at this and make some conclusions like, oh, mold 2 didn't do as well as mold 1, but then in competition, mold 2 did much better. Why is that? That's when you start making some hypotheses and writing in your discussions and results. Okay, now you can do the same thing with your treatment data, right? You can plot mold 1, which is this line dot dot line dot dot here, mold 1 without competition, but with treatment, so with, say, Lysol pH again, whatever. Then you've got mold 2 here. Then you've got mold 1 with competition and mold 2 with competition. This purple line is your no mold plates. That should be 0. If you've got contamination, it'll look different. Okay. Things to keep in mind here are that my axes are the same, 100%, 100%. The days are the same. Titles, axes, labels, keys. Also, I'm keeping my keys consistent. So mold 1 is always in black. And depending on whether it has treatment or is in competition or not in competition, it's got a different style for the line. So mold 1 in competition and control is this dotted line. Mold 1 in, not in competition in the control group is a solid line. Solid line is also control without competition, but it's in red for mold 2. So you see how this is consistent. That's pretty important for making this easy to interpret the data. Now if you look at this graph, it's kind of confusing. I mean, this is an easy way to see how treatment affects mold growth and competition. But that's not really our fundamental question. Our fundamental question is, how does our treatment affect mold growth, normal mold growth, and normal mold competition? So really we're asking a question about treatment effect in comparison to the control growth, which is why we did our controls in the first place. So you really want to be going for graphs like these, which compare treatment and control. So you see here I've plotted mold 1 control, same line as this line, with the solid black for mold 1 without competition. You can see it's flat line from 6 to 7, same flat line from 6 to 7, right? I've just taken this exact line and transposed it here, okay? Same for mold 2, copy this line, put it here, and now take the line dot dot data, basically the mold growth without competition data from the treatment group, and put it on here so that we can see directly how the treatment affects the growth of the molds alone in their own plates. And again, that's this line, just copy and paste it over here, and this line here, copy and paste it right to here. And you can see from this data that there's not too big of a difference between mold 1, between control and treated, but there's a pretty big difference between mold 2, control, and treated. That's something to talk about in results again. Now over here, we've done mold growth. That's what I'm calling the mold's growth without competition, but we want to also look at how the treatment, say applying Lysol, affects the molds in competition with each other. And that's this graph here. So we're taking the dotted lines from the control data. That's our mold composition baseline. And we're going to compare it to how the molds competed in the presence of our treatment group, or treatment effect. Okay, and we plot those things together and we can see, man, those track really closely to each other. So there's almost no significant effect on competition between molds 1 here, these two lines, and molds 2. So basically this treatment, it did not affect mold competition. It didn't shift which mold won quote unquote, or which mold competed better for resources. But it did change the individual mold growth, and that's a really interesting finding in that the treatment does affect the mold individually, but it does not affect the mold in competition. So that's something you want to be writing about in your discussions and results sections if that is the case for your data. Anyway, I hope this video is helpful to you guys. I know this stuff can be confusing. It's a lot of data, and this is a really important part of your lab grade. I just want to make sure that you guys are getting the most out of it, and also that you guys do well on your reports. So take this into consideration. I hope this helps. Again, you want to be comparing treatment to control. That's why we're doing these graphs. So these are sort of the most important, but you can do all four of these. This one also tells you something, but these three are the most important. Okay, and I should have this data from everybody soon, and I'll be putting that up so that you can get this data to make your averages. Okay. All right, I'll see you in class, and good luck.